investigation is a height of ignorance. With these words of Albert Einstein and amid the auspicious Navratri festival, I, Dr. Falguni Joshi, welcome you all to this webinar. Clinical lab testing plays an important role in delivery of quality healthcare. At the same time, informed decisions can be made as to the most appropriate treatment for the patient. And this has been proved in the current pandemic situation. But at the same time, we are surrounded by a lot of misinformation regarding the vaccine, which is supposed to be a critical to the ending of COVID-19 pandemic. To give us an insight in the investigation of COVID-19 condition, as well as the clear view about the myths and facts of vaccinations, we have with us Dr. Suvarna Joshi, Madam. Madam is an associate professor at the BJ Government Medical College in the Department of Microbiology. Madam is currently working as in charge of COVID-19 diagnostic lab approved by ICMR. She is an ex-secretary of Indian Association of Medical Microbiology Maharashtra chapter and an ex-secretary of the Research Society of BJ Medical College and Sassoon Hospital, Pune. Madam has penned down more than 25 publications in national and international journals and has presented more than 50 papers and posters with winning the prizes. She is a recipient of the Fogarty Fellowship by Johns Hopkins University, USA, and a recipient of UNESCO American Society of Microbiology Award. We welcome you, Madam, to this Dr. G.D. Paul Foundation, YMT Homeopathic Medical College. Kindly guide us through this vaccinations and investigations.
Thank you, Madam, for your kind words. And in the first, let me thank the G.D. Paul Foundation and Principal Page Madam and Vice Principal Sir, and also you and for coordination, Dr. Pallavi. You have taken a lot of efforts to coordinate with me. And with this small thanks, word of thanks, we will start the session. Shall I share my slides, Madam?
Yes, Madam. Yes. You can directly.
I can do that. No?
Yes, yes.
So good morning, everyone. Today I will be discussing and giving you a little overview about the laboratory diagnosis of COVID-19. Many of us are aware about how we go for COVID diagnosis, but I would like to share the microbiological aspect as such. This is not the first so-called pandemic which we are faced. A few years back, we have faced the H1N1 swine flu, and before that in 2005, we have experienced the SARS also, right? But the amount of population involved or get infected was less compared to the COVID, as we all know. But the major difference in the diagnosis is that the first test, which this is the first time in the history of any pandemic that the whole world is going for diagnosis using a single kind of a test, and that is the RT-PCR test. So I would like to give you a little bit information about how it is possible to go for a diagnosis of COVID-19 by PCR as such. Because even though we had faced the HIV in way back in '83, that's the time it has been identified first etiology as a viral etiology, but still diagnosis was not solely based on the RT-PCR. But in this case of COVID-19, it was possible because of these three associations. One is GSED, pangolin, and next-gen. Now, these are the three associations. This GSED is the global initiative of sharing all information about influenza data. They have the data about full genome sequenced so far all over the world. And this is the website picture I'm showing you. So there are about 4 lakhs genome so far being sequenced and uploaded in their database. And because of this initiative, which was already there, it was possible to design the primers for rapid diagnosis by RT-PCR test. And this is the first pandemic where almost whole world is now trained. The lab people are trained to go for lab diagnosis by doing the RT-PCR test, and the main efforts went through this because of this GSED association. Then something I want to share is the structure of the virus. This is not, as I know, I'm not taking an undergraduate class, but this I want to show you because this is the each structure which is coded by individual gene, say the spike protein. This is being this thing by coded by the S gene. The membrane is coded by the made up of M protein and is coded by M gene. The envelope is coded by N gene. So that E, M, S, these are the genes we are making fragment of that to use as a primer and go for diagnosis of COVID-19. So that's the reason I'm just showing you where it is situated. These knob-like structures are nothing but the spike protein spikes, with the help of which the virus is attached to the human cells on the ACE2 receptors. Now, this is one of the biggest virus lengthwise. If you see, it is RNA virus and it is one of the largest genome among the RNA virus family. The genome length is about 29,903 base pair in length, and there are it codes for about 29 proteins. But for production or for synthesis of this protein, they need the help of the human host machinery. Okay, so this is the classification of coronavirus. I'm not showing you as a microbiologist, but I just wanted to put a light on the thing that it's not the first time the human being get exposed to the coronavirus or acquired coronavirus infection. Before that also, the human being has exposed themselves or experienced the corona infection. Say, for example, as I said, in 2005, we had experienced SARS-CoV, severe acute respiratory syndrome virus infection. Then just 2 years before, we had the Middle East respiratory syndrome virus. Okay, then in Hong Kong also there was human coronavirus outbreak, so on and so forth. So this is not the first time that coronavirus infected the human being. Okay. Now, in the newspaper, you must be reading about the VOC, VOI. So what are these VOC, VOI? So these are the nomenclature or classification proposed by the US government. So US government has their team, which is known as US SARS government SARS-CoV-2 Interagency, SIG. This committee has proposed the corona. So far, how many different subtypes or variants or mutants have been identified of coronavirus? They have been divided into 3 different classes. One is variant of interest, variant of concern, and variant of high consequences. Now, when to say that it is variant of interest and when to say that it is VOC? So when the virus subtype or the mutant is been identified for the first time, we call it as a variant of interest. But when the proportion of infection by this new variant, new mutant, goes about more than 20% of the total positive in that particular area, we call it as a variant of concern for that area. So variant of concern for India may not be the variant of concern for UK or US, because the mutant which is prevalent in India may not be prevalent in US and UK or Belgium or something like that. So it is geographical area related, based on how much percentage of positive people harbor that particular mutant. Okay. And when it causes the severe clinical manifestation, death, morbidity, and mortality increases, then it becomes the variant of high consequences. Today, there is no variant of high consequences. Initially, there are some strains which are being first time identified in UK, and that is known as alpha. So in a different area of the world, different variants or the mutants has been identified. So to remove the confusion, this US government has defined them as a VOC, VOI and high consequences variant. But in September, they come up like there is a very difficulty in or a thin line when to say VOC in this area and that area, and the media people is also causing confusion. So they removed this and they came up only with single terminology, that is variant being monitored. Okay. So routinely we identify the mutant with lineages numbers, say beta lineage, so it is a B.1.1. So it is difficult to understand by the layman. So World Health Organization came up with the Latin alphabet system for classification of this different, or the nomenclature for the different mutants which are prevalent in different parts of the world. Okay, say for example, the alpha variant is common in UK and first time identified in UK. Similarly, beta variant was first time identified in South Africa, gamma in Brazil, zeta was also first time identified in Brazil, lambda was also identified first time in the Brazil, and in India, delta was the one which has been first time identified in India. And today the scenario is that all over the world, delta is the most common variant causing infection all over the world. Okay. So once I say it is alpha, beta, gamma, then it becomes easy to understand like which is the infecting strain. But if I say it is B.1.617.2, then everybody will look at me, what she is talking. But when I say delta is isolated, then everybody can understand. So that's the easy way to understand which mutant is causing infection. So why do we need to know which mutant is causing infection? It is important to know because the different variant has a different clinical severity. So with identification by genetic sequencing or genome sequencing, you can make out like whether the further wave will be responsible for more death. Is it necessary? What necessary precautions you need to take, and so on and so forth. So this is the first time in the pandemic, the whole world is going for the sequencing. Now, there is one more association which is known as pangolin. So this pangolin stands for phylogenetic assignment of names to the global, to the influenza virus, the COVID virus, which is causing global outbreak. Okay, so this is the association which gives the nomenclature. And there is one more association, that is next strain. These are the people who are working on bioinformatics. And on the bioinformatics, on the basis of the bioinformatics, they are able to track the evolution of COVID-19 virus. So all the computer and artificial intelligence, bioinformatics, everybody played their role to take a control on this pandemic. Okay. So this is the slide I am showing you just to show that these red spots there are some red spots, there are some green spot in the brain area you can see, and there are some blue one. These are the spots which represents the receptors for at which the COVID-19 virus can attach. So you can see the almost whole lung is susceptible for this COVID-19. So that's the reason why the lung is, or the respiratory system is the one which is first get affected or more get affected once the virus is inhaled. Now a little bit about which chemical agents or which disinfectant one should use. Okay, it's a sensitive, being a virus, it is sensitive, but since it's an enveloped one, it is comparatively resistant as compared with the non-enveloped virus. Okay, but so-called whatever daily disinfectants which we are using, it is sensitive to that almost all the disinfectants, such as ethanol, peracetic acid, sodium hypochlorite, and everything. Only thing is that it is resistant to the chlorhexidine, that is Dettol. So when you are selecting the disinfectant, be careful. Do not select the Dettol products or chlorhexidine based products as a disinfectant, because virus may not get killed easily with the help of this. Okay. It is sensitive to the heat. As you go on increasing the temperature, the virus will get killed in a shortest duration of time. Say at 50 degree, within 30 minutes, virus will get killed. But if you increase the temperature to 70 degree in 5 minutes, it will get killed. So what I want to stress, like when you are doing the home quarantine for your own family member, now how to get rid of the virus from the clothing and everything. So just put it in a boiling water with soap, and that will take care of it. So nothing to worry ki आता मी हे सगळं फेकून द्यायला पाहिजे का. Nothing, no need to do that. Okay, simple soap and water is enough to kill the virus. Remember, it can remain viable on the plastic surface, glass surface. So all the surfaces you can clean it with the disinfectant and get rid of virus. Now, when you are collecting the sample and sending the information, filling up the patient's detail, you must be wondering why you are showing it to us. So basically, when you are collecting the sample or when you are giving the sample, this is the specimen referral form which ICMR has said like one has to fill up along with the form and submit it to the lab. Okay, so in this form, there are some mandatory information regarding the address, contact number, age of the patient, name of the patient, and the symptoms. Why? What my sincere request is that give all the detailed information which you have when you are taking the sample as a patient from the patient, or you are giving sample as a patient. Why? Because all this information we are putting it on the ICMR website. Each and every field which is being filled up by the information given by the patient, the lab people are uploading it on the ICMR website. Okay, so if you do not give the information, whatever the data is going to ICMR, they are analyzing it and the wrong information will be come out. So for example, just simple example I am giving you. Now, since we have started vaccination from almost mid of the January, 16th Jan was the first day where the vaccination started in Maharashtra. So from January onwards, now it is almost October mid. So maximum people, mainly the healthcare workers and frontline workers, are vaccinated. But when they give the sample, when they collect the sample, hardly any information regarding the vaccine details are coming or vaccination details are coming to us. So we cannot comment on how many people, even though they get vaccine, is getting infected again and not getting protection by the vaccine. So today, if you ask me, the figure is say 0.0004 or something like that. But is it the real figure? No. I will just give you one example. In the month of June, we had from one particular center about 110 samples, and then out of that, some 11 were positive, and that was not the peak time where you have a more than 8% positivity. So I was wondering why so many people in one center at particular day came positive. So I inquired with the medical officer, is it because by the name when I look at it, it was not a family member also. So I contacted the medical officer and asked, is it from some particular area? Because the address wise also they are from different area. So then he said, I will find it out and I'll let you know, madam. But I could not keep quiet. So I contacted those positive patients and found out what is happening, like why so many people are there and is they are connected anyway. So then I found out like they are all connected. They were from single police station. One of their colleague came positive. So the police inspector in charge asked to get them, all of them tested. So out of how many of them, almost 100 plus were from that police station, tested themselves and 10 were positive. Out of those 10 positives, 7 were vaccinated. So can you imagine? And none of them gave the details like whether they are vaccinated when this form was filled up. So that is the reason why I am tracing it. Please give the information. If you don't give the information, all the wrong conclusions are coming up. So these are the material used for sample collection. There is one thin swab and there is one thick swab. So you must be wondering why we must know what is to be done with this. So this thin swab, use it for nasopharyngeal sample collection, and the thick swab, use it for the oropharyngeal sample collection, because otherwise it may get break if the thin one you are using it, and there may be a problem to remove that broken swab from the esophagus. Okay. Now there is a difference in the length of the swab stick and the VTM tube. This is the viral transport medium tube. So there is a difference in the length of the tube and the length of the swab. So after collection, it is expected that you will cut the extra length of the swab stick. Most of the time, what the collecting technician does, he just bend the swab stick and put it in the VTM and tightly screw cap the lid. But when we open it in the lab, if it is bended like this, the moment we open the cap, that stick just go up like a dart, and that sample gets spilled in the biosafety hood. So to avoid such accident, it is recommended that one should cut that length of that extra length of the stick. And then make sure that sample will be transported in the polythene bag. So in case if it is leaked out, it will not contaminate the all other samples which has been transported at the same time. Only that particular leaked out material or the swab can be taken outside. Okay. And samples should be transported at 4 degrees. If it is not been transported at 4 degrees, there is a chance, like if the sample is having low viral load or the low viral number, and you are transported the sample at room temperature, then there might be the degradation of the virus may get killed, and the viral RNA viral genome will get degraded, and we will be reporting it as a false negative. So to avoid that false negative, it is recommended that the sample, once it is collected, should be kept in the refrigerator and should be transported at 4 degrees. Now, who should be tested? Because now there is a season of common flu also. One has to be very careful. Just don't ignore that it can be a common flu. Mala darvarshis yeto. There is no necessity to test it. No, do not do it, because even though you can tolerate, your body can tolerate, you are the one who will be acting as a source of infection for the community. So once you have a symptoms, test yourself whether you are having COVID infection. Okay. So once you go for in the lab for the sample, then it is done. There are tests which are being done routinely, which is fast, is the antigen testing. First, so the swab first will be collected for the antigen testing, and then if the antigen test come negative, then only we collect the swab for the RT-PCR. Okay. Once the antigen, that is a rat test, is positive, it is recommended not to collect the swab for RT-PCR, because once it is positive, it is positive. Doesn't matter whether it is positive by antigen test or whether it is positive by RT-PCR test. Once positive is always positive. So no need to confirm the antigen test results by the RT-PCR test results. So do not insist कि मी antigen positive आले पण मला बघायचे तो false positive आले का, so let me go and do the RT-PCR also. या lab चे results reliable नाही. Wherever you are going, antigen test is, if it is coming positive, that means you have a high viral number in the nose or in the oropharyngeal cavity, and that's the reason your antigen test is positive. So no need to go for the RT-PCR test. So for traveling, nowadays it is mandatory to do the testing. Now, when the government has made it mandatory that you should be doing testing before you travel and you should have a negative report, the reason behind that, you will be traveling, you will be sharing that travel, whether it is a bus or the railway or the air bus, along with the other passengers. And if you have are positive, then in those duration of travel, say 5 hours, 6 hours, you will be infecting. There is a chance like you will be infecting the surrounding passengers. So it is recommended that you should, before you travel, you should have a negative report in your hand. So do not insist that I should get a negative report. If it is positive, you will get a positive report. And it is not only for you, it is for the community. You have to isolate yourself, you have to home quarantine yourself, because you will be acting as a source of infection for the community. So please follow the guidelines, and if you at all you have a positive report, do not travel along with the positive report, or do not hide your positive report and travel it. So the tests which are available currently for diagnosis of COVID-19 is the rapid antigen test. Then there is RT-PCR test. In the RT-PCR test, there is a conventional RT-PCR test and there is a rapid RT-PCR test. So one of the rapid RT-PCR test is the CBNAT, or the gene expert test, just like a tuberculosis. This CBNAT, we are using it over the years for diagnosis of tuberculosis. The same company has come up with the kit for diagnosis of COVID. And within 50 minutes, the sample is analyzed by PCR and the reports are available whether the patient or whether the sample contains COVID RNA or do not have the COVID RNA. Okay. So the rapid antigen test, that is the immunochromatographic test, is available. Usually the care has to be taken, like once you have taken the sample for antigen testing, say from one nostril, then, and if it comes negative, then take the swab from another nostril for RT-PCR. Do not take the sample from the same nostril, because it is like wiping the table. First time when you have put the swab, you have swipe out, you have clean up that area. And there is no enough virus to give you positive result by rapid antigen test. Second time when you are putting the swab in the same nostril, there are still less now, because whatever are there, they had come up with the first swab. When you are putting the second swab in the same nostril, again there is a less one number, and that is, the number is so less that it will not be able to pick up even by RT-PCR. So it is always recommended that collect the swab from two different nostril or from two different areas. So you can decide whether to take one sample for nasopharyngeal sample for antigen testing and oropharyngeal sample for the PCR. Like that you can do that, but do not use the same area for collection of sample for antigen and RT-PCR test. And remember that blood sample is not used for, we are not using the sample for RT, even for RT-PCR and for antigen testing. Because many people have this concept that antigen test करायची आहे का मग blood sample दिया. But for this COVID-19, the sample which is collected for antigen is also the swab, and for PCR is also, it is a nasopharyngeal or the oropharyngeal swab. So this is the way the rapid antigen test is being done. The swab is collected, nasopharyngeal swab, and then it is put in a lysis buffer. So whatever number of viruses which are present, the envelope will get lysed out and the viral RNA will come out. And then you have to put the drop of this lysis buffer on the cassette and wait for 15 minutes for development of the color. So like these cassettes are there, and you have to add that lysis buffer in this well, and it will flow laterally. So it is also known as lateral flow. Okay, so you will have one test line, and if the sample is positive, you will have a test line. Otherwise, only control line will be developed. This antigen test is the cheaper and the fastest or the rapid method, and the results are available within 15 minutes. If patient is positive, within 15 minutes, you get the report. No sophisticated instrument is required. No training, much training is required. No technical expertise is required, and the test can be done bedside. So that's the reason the MyLab company has come up with the home package or the self-testing kit, which they had come up, brought in the market. So it's the same thing is there. You can collect, they give you the instructions how to collect the swab and how to do this test. And after you do the test, they give you the on the app to log in and give your details of your own, the age and address and this thing, and the kit, the app directly upload information on the ICMR website. Because it is mandatory, whosoever turns out positive or whosoever is tested with because he or she has symptoms, has to be registered, has the data has to be entered, like these many suspected cases are tested, and out of that these many came positive and these many are negative. So every test has to be recorded. So the second test, this is the one which I was talking about. This is the cartridge-based nucleic acid amplification test, or the CBNAT. This is the machine or the instrument, and this blue one is called as cartridge. So you just have to open this blue lid and add that sample, that is the viral transport medium in which the swab is collected. Okay, and just put it in this machine like this and close this. And within 50 minutes, it is attached to the computer. The software is running and taking the signal and gives you the result within 50 minutes. So this is the machine. So at a time in this machine, you can load 4. Like that, we have a 16 cartridge at a time we can put it like that machine. This 4 cartridge machine, we had kept it in the casualty. So if the serious patients comes to the casualty and he is like suspected to be of COVID, we immediately process his sample or her sample in this CBNAT machine, and within a 1 hour time, we give them the report whether the patient is CBNAT positive or the COVID positive or COVID negative. Because we can do this rapid reporting, within 1 hour, the patient can be segregated into the COVID or non-COVID ICU, and the life can be saved. So it is the fastest RT-PCR test, and within 50 minutes, the results are available. And then we'll come to the conventional RT-PCR test. So those who are stable patients, antigen negative, but symptom wise there may be the they are the suspected case of COVID. In such cases, we are collecting the sample for the conventional RT-PCR test. Okay, so the sample, once it comes to the lab, we open up it in this biosafety hood. Okay, you can see these perforations over here. Through this perforation, the airflow is coming up, and on the top of this biosafety hood, there is a HEPA filter. So whatever air is coming from the what you can say base of the biosafety hood, it has a so-called current. So if any droplets are generated in this biosafety hood, that airflow takes it up, and there is a HEPA filter, so it gets filtered out, and whatever air coming out of the biosafety hood is clean. So up till now, we have processed almost 5,94,000 samples in our lab, but none of the person who is working in the lab got infection because he is working in the lab. So that much precautions we are taking, because every sample we open up in the biosafety hood. So usually, what are the steps which we are doing it in the PCR? So initially we used to take 8 to almost 20 hours, because we used to do everything manually. So whatever VTM or the viral transport medium, the sample which we have received, we are doing the extraction of RNA by various method, manual method or automated system. Initially we had only automated, we do not have the automated, and manual system has to be used. So it used to take us 2 hours for 12 samples. So with that speed we used to go. And then once you get the RNA from the sample, we put it PCR, and then PCR takes about 2 hours, and then a total like that 6 to 7 hours we take it. In the PCR, we amplify the RNA and find out whether the sample contains viral RNA. Okay. So this is the hood which I have shown you. This is the automated nucleic acid extractor. You can see there are some plates like ELISA plates. So at a time, 96 samples can be put in this machine for extraction of RNA. And this machine takes half an hour for extraction of RNA from 96 samples. And in contrast to that, when we were doing it manually, it was only 2 hours we used to take for 12 samples. So the speed, you can imagine, we can achieve because of this automated nucleic extractor. We can do now even 4,000 samples in a day. Otherwise, it was a nightmare for us to even process 700 samples manually. And the next machine, this is the thermal cycler, where the amplification is going on, and it is known as real-time polymerase chain reaction, or the reverse transcriptase polymerase chain reaction. Okay, now those you must be hearing about for the positive sample, those which are the positive sample reports, you must be seeing the CT value. So I will just tell you what are these CT value means. Okay, so you in the amplification process or in the RT-PCR process, we carry out the 40 cycles. So what 40 cycles we are doing? We are taking the sample or the RNA along with the other chemicals required for the PCR test. We take that sample first to the machine, goes up to 95 degrees. Okay, so whatever sample has the DNA and this thing, it comes to the single standard. And then in the reverse transcriptase test, what happens? First time we are doing conversion of RNA into the DNA. Okay, that is the reason, with the help of the enzyme reverse transcriptase. So that's the reason the test is known as RT-PCR. So then the next step, taking all that sample which are now the RNA has it will get converted into the DNA, and then that DNA we are taking it at 95 degrees. So the double stranded DNA gets converted into the single stranded. Okay, and then for both separated out single stranded of DNA, the complementary primers go and bind, and they synthesize the complementary strand for each of that separated DNA strand. So from two strand, at the end of first cycle, you will get 4. And like that, if you have initially 10 copies of a single stranded DNA, you will have the 20 copies at the end of first cycle. So like that, almost 10 raised to 30 amplification. This is, we call it as an amplification. So whatever number you have in the beginning of the cycle, it becomes double at the end of the cycle. So like that, 40 cycles we carry out, and this is known as amplification process. So like that, 40 cycles we carry out, and we see like this curve at the end. Okay, so when we say that this particular sample has a CT value 25, means at the cycle number 25th, the detectable RNA can be seen in the form of sigmoidal curve. This is called as sigmoidal curve. And in the RT-PCR test, we are using two different genes, either S gene or N gene or E gene, like that. Okay, so that's the reason for every sample in the report which are positive, you will get the two different CT values. So when we say that CT value is 25, means at 25th cycle out of 40 cycles, detectable quantity of RNA is seen. That is the meaning of CT value 25. Okay. So in the beginning also I have shown you this viral structure. So these are the different genes which we make use of for the primer preparation in the PCR. So E gene, ORF gene, S gene, N gene, or RDRP gene. These are the specific gene. These ORF, S, N, and RDRP are the specific gene for COVID-19. And E gene is common for all beta family of coronavirus. Okay, so usually the kit contains E gene and one of these 4 genes. So first we confirm that whether whatever RNA we are amplified is from the beta corona family, and then once it is detected as beta family member, then we see whether it is the SARS-CoV-2. Okay, so the E gene plus one of the 4 genes are required to say that it's a positive sample. Plus the amplification, detectable amplification should be seen before 35th cycle. If amplification is seen after 35th cycle, we do not report it as positive. So if a CT value is more than 36, we report it as negative. Now, some time we have to give report as inconclusive when the CT value is between 35 to 36 for one gene, and for other gene it is less than 35. So it's not clear-cut negative, neither it is clear-cut positive. So in such case, we give report as inconclusive or presumptive positive. And it is expected that such samples, samples from such patients should be repeated after 48 hours. And it has been proved that such, if the sample is collected at the early stage of the clinical course of infection, after 48 hours, when the sample is collected again, these patients come positive. So whenever you get an inconclusive report, repeat sample. Okay, so this is about the RT-PCR test and the CT values. Now, even the antibody testings are kits are now available in the market. Now, when to use, what is its use, and why it is not being routinely used for diagnosis. Little bit about that we will see. Just like antigen kit, immunochromatography test for antibody, IgG as well as IgM antibody test kit is available. But there is no clear data available that whether the patient will develop an IgM antibody. As we know, like for following any infection, the first antibody response which is shown by the human being is IgM antibody. But in this case, it has been seen that some patients show IgM as well as IgG at the first time. It's not like first IgM and then IgG, which is you routinely seen in many infections. So then that becomes a problem. If it is only IgG positive, what to do? If it IgM positive and clinically symptomatic, you can give the report. But if it is IgG, then whether it is because of the past infection, still he is having IgG, or current scenario clinical manifestations is because of IgG, we have no enough data, and that's the reason why this test is not being used for diagnosis. Okay, so we are using it for the serosurveillance. It can, in the pre-vaccination era, it has been used for the identification of the hidden cases, especially when because of the social stigma in the first phase of pandemic in last year, people used to hide because the full family असं वाळीत टाकल्यासारखं करत होते. So not to tell to the others. So in that situation, what used to happen, other family members are also getting exposed to the infected person, and they may also had a subclinical infection. So how to identify such hidden cases? So in such family members, we have seen like they have shown the IgM antibodies. So we can use them. We had used them for the identification of the hidden cases also. It has been seen that both IgM and IgG has appeared within 7 to 15 days. But more studies are required to find out the exact timeline and its utility. We are still using it for the plasma therapy also, because this plasma therapy, when it has been trial has been started, then that's the time they had said that once person is recovered from infection in his convalescent phase, you test his plasma for IgM and IgG, and if only IgG antibodies are there, then only you take him for as a donor. Okay, but what has happened that the titer should be minimum 1:640. But there are no enough kits available for the quantity or the quantitative test for antibody. So there may be a chance that those plasma which has been given for certain individual may not have enough antibody, and that's the reason the therapy may have failed. But now the kits are available, and it has been also observed that if person is vaccinated and he has also contracted the infection, then large quantity of antibodies are produced by such individuals, and then that might be the very good plasma for plasma therapy. Okay, now this is the that is about the antibody this thing. Now there is one more thing which I want to tell you, that there are people or the labs which say that we have the kit which can differentiate whether you have an antibody and whether they are from the after vaccination, or whether they are after because of the normal infection or natural infection. But nothing like that. The kits which are available are against this S protein. So virus attached to the human cell with this spike. Okay, and these are the receptors, ACE2 receptor. So virus come and binds to this ACE receptor, and then it is being taken up by endocytosis in the within the cell, and then it is being the RNA is released, and then the replication takes place, and again the virus gets reorganized and then produce its own viral copy, and the progeny comes out. So what I want to say that this is the spike protein with the help of which the virus enters the human body. So if you can block its attachment to the ACE2 receptor, the further whole episode can be blocked. And that's the reason why the vaccine is prepared against the S protein, or the Covishield which is available is based on this S protein, because it antibody produced against the spike protein can block it, the further sequence of the event. Now a little bit about the vaccine. There are different types of vaccines which are available. There is something called as protein subunit vaccine. Just like for hepatitis B, we have the subunit vaccine. Similarly, we have the protein subunit vaccine. It is being marketed by the Novavax. Then the S gene, which is incorporated into the non-pathogenic virus, and that is the Covishield. So it is a viral vector vaccine. The virus which is acting as a vehicle or as a vector for transporting the COVID genome into the human body is non-pathogenic to the human being, but it can replicate and it can give the COVID viral genome into the human body, and then that can be translated into the antigen, and antibodies can be produced against that antigen. Then there is something called as mRNA, which is being developed by the Moderna and Pfizer. Then there is plasmid DNA vaccine, which is coming up by the Zydus Cadila, and the Covaxin, which is the whole virus inactivated vaccine. Now there is a lot of debate, which is a good one, which is a bad one. All are good ones. There is no this thing like this vaccine develops 70% antibody and this vaccine develop 80% antibody. Everybody develops enough and the same quantity of antibody. The antibody production or immune response towards antigen or towards vaccine is solely depends on individual's immune system. Okay. Now there is a thing that whether the vaccine is to be taken or not, and whether there is any side effects and which one should be better, whether it is a killed vaccine is better or whether it is a live vaccine, this Covaxin is better or Covishield is better. But it has been observed that both has a equal efficacy when the clinical trials are being done. It has been found that it has an equal efficacy. Okay, now there is one more question in the individual people's mind, that when I taken the vaccine, why should I get the infection? So remember, you have taken the vaccine which is intramuscular. Okay, so virus is coming through the nose, and in the nose there are no antibodies even though you take the vaccine. So virus is bound to enter the human body, but if you observe those who contracted infection even after vaccination are almost asymptomatic or has very mild clinical features or the symptoms. So the vaccination has reduced the morbidity, has reduced the clinical severity. So it is necessary to take the vaccine. The second thing, like when why the vaccine cannot prevent further infection. It will prevent further infection when you can block the virus at the entry point. So remember, do you remember when the vaccine was available for swine flu? It was a nasal spray, nasal drop. And because of that nasal spray, the local IgA antibodies has produced, and that has helped in blocking the virus at the entry point, at inhalation point. And that's how the swine flu was controlled. So similar kind of nasal vaccine trials are also going on, but not a single vaccine yet come in the market. And that's the reason local entry point has not been blocked by the vaccination or by the antibodies. That's the reason even after vaccination, there is a chance like person may get infection. Now there are a certain terminology like what we call it as a vaccine breakthrough. like individual after taking the second dose of vaccine, 2 weeks after, if he gets the infection, then we call it as a vaccine breakthrough cases. We have experienced, we have seen certain cases which are vaccine breakthrough cases, but whether to call it as a vaccine breakthrough case is really a question mark, in the sense like those individuals who are contracted the infection after vaccination did not develop antibodies after vaccination. There are many cases which we have seen even after first dose, even after second dose, they have not developed the antibodies and they had contracted the infection. So whether to really call them as a vaccine breakthrough case is a question mark. So it is important to take the vaccine because it will develop the protection, it will develop the immunity against SARS-CoV-2. It has been seen that it has reduced the mortality, reduced the clinical severity, as well as reduced the hospitalization. Now, even though Sassoon Hospital is a 550 bedded hospital for COVID itself, it used to be totally occupied in the first phase and even in the second phase in this year, March, April. But now we had hardly 20-30 cases, because it's now 7 months that vaccination is going on and the people has developed immunity. So hospitalization has reduced drastically. Plasma therapy I talked about, and these are the different kind of antibody tests which are available. Rapid test like antigen test I had shown, the chemiluminescence CLIA test is there, the ELISA test is also available, and nowadays what they are marketing is the neutralizing antibody test. Now IgM, IgG or IgA, whichever is the type of antibody, almost all of them has a function to kill the virus, and that we call it as a neutralize the virus. So in the neutralization test, we see its capacity of these antibodies, whether they are capable of neutralizing. And that is the test, what we call it as a neutralizing antibody test. So that kits are now also available. So after vaccination, whether you can see the neutralizing antibody, enough antibodies are produced, and whether functional antibodies are produced, that can also be checked. But do not get upset. I have taken the vaccine in the month of Jan, and now when I tested in the month of October, I am not having any antibody. Though you do not have a detectable antibody, you have a memory. You have a memory cell. Your immune system has remembered when you took the vaccine. This type of antigen I had came across, and if it comes again, I must attack it in a rapid action. So the memory cell will act fast and you will not suffer from the severe clinical manifestation. So do not get afraid if you do not have antibodies after 8 months or after 6 months and so on. What we have done just last week, about 1,000 samples we have tested, individuals we have tested, and we have seen in after 6 months, almost 99% of individuals have shown presence of antibodies. So nothing to worry. If you have taken vaccine, you are having the protection. So that's all from my side about the vaccination and about the lab diagnosis of COVID-19. Thank you so much for the patience, viewers.
Thank you, ma'am.
Thank you.
Thank you so much for enlightening us. And it was very informative session. Thank you so much, ma'am. Now we are having so many questions. So
Okay.
can we take a few questions?
Yes, yes,
Okay.
madam, please.
So first question is regarding disinfection of mask. Ma'am, some people use cloth mask. Is it right to use cloth mask, and how to disinfect the cloth mask?
Yes, it is. There is no harm in using the cloth mask. You can just put it in a, as I said, in a boiling water with soap and wash it. You can keep 2 every day. You can wash it and use it. No harm in doing that.
Okay. Then next question is regarding HRCT. What are the indications for HRCT? When we can do the HRCT test?
When you nowadays everybody is aware that they should keep the monitoring the oxygen level. Oximeter has to be there. So when the oxygen saturation goes beyond below 90, one has to test themselves
Okay.
to
Then
see whether. But remember that HRCT or is not that whatever the ground glass opacity you are seeing it may not be always because of the COVID. So what we say that these are the supportive diagnostic tests. Radiology is always supportive because many times people have experienced that they have the picture ground on HRCT score is 10, 15 and the PCR test is negative. Then whether to call it as a COVID or not. So whether to call it as a COVID or non-COVID, the treatment is same. Only thing is that unless and until you have a clinical diagnosis confirmed by this thing, only then the patient is shifted to the COVID. Otherwise, he remains in the suspected COVID ward. Okay.
Then next question. A patient with CT value 16, but the patient is asymptomatic. And then what we should do? Is it convenient to start with Fafnavir? Though the patient is asymptomatic, his family member is having symptoms. So can we start with the medicine directly?
If person who is having HRCT score 16 and not having symptoms, then he has to be just. But other family members is having symptoms, then that person who is having symptom will be treated symptomatically. And that family member should be tested whether he is COVID positive also. Okay. And if the family member who is having symptoms and is COVID positive, based on his own CT score, he can be treated. But unless he is having symptoms and not having symptoms and HRCT score 16, he should be kept under observation and monitor regularly after 6 hoursly his oxygen saturation.
Okay. Then next is patient taking COVID vaccines are coming with the other symptoms like arthritis and all. So can you guide us regarding that? Have you observed these kind of symptoms?
There are no such cases. At least we have observed, like after taking vaccination, they had come up with arthritis. It is maybe their own previous old or the chronic arthritis, and they may have they are trying to correlate it with the vaccination status, because many people in their mind has a stigma or the fear, and they are trying to associate it with that. A day before that only, one of our colleagues, family member, they took the vaccine. The girl is only 18 years. And once she took the vaccine in the society itself, and she fainted. And then the people were making chaos. Ki, are, chakkar yeun padli, in conversion after vaccination. Nothing was that. She was so scared of taking injection. And because of that, she fainted. So it is nothing to relate with the vaccine as such. Content of the vaccine has not been shown any response like these symptoms. But always there is a chance who is having autoimmune type of a disorder can show the exaggerated symptoms.
Then next question.
Yes.
There are certain mouth sprays available in market. They contain enzymes. So they claim that they are required for, they will reduce the viral load. How effective are they, and are they reliable? Mouth sprays.
They must be reliable. But if you ask me, there is no necessity. If you are using the mask, there is no necessity to go for the mouth spray as such. The simple thing what you can do is just do the gargle with the hot water. Because why exposing unnecessary with the chemicals? Even once you come at home and just do hand washing with the normal soap and water, that should be enough. No need to take the mouth spray as such. Any other?
Okay. I like all the students, if they are having any queries, they can type in chat box.
Yes, sure.
Ma'am, can I ask one?
Yes.
Dr.
Yes.
Uttara here. Ma'am, what is the importance of D-dimer testing in COVID protocol?
Okay. This D-dimer testing is to see the what we can say hematological disorders as such, the bleeding disorder. And it has been seen that those patients who have the COVID and exaggerated immune response, so they land up in the these hematological disorders, and that can be monitored with the help of D-dimer levels. If the D-dimer levels high, then that means the patient has a bleeding tendency. So clot formation can occur anywhere within the body, and that can cause the harm. So to do the monitoring of that, the people do the D-dimer.
Ma'am, is it necessary that once RT-PCR, with RT-PCR, if it is diagnosed, then D-dimer is necessary to be done in the protocol?
It is depend on like the patient's other comorbid condition. So patient is 20 years old and he is healthy and hearty, very mild symptoms are there. There is no necessity. But you know, nowadays there is a so-called lab test package. So that includes all these tests.
Okay. Okay. Thank
So,
you so much.
and nobody wants to take any risk. So एक करून टाकूयात आपण, बेसलाईन माहिती असतं. अदरवाइज आयुष्यात आपण कधीच D-dimer केलेली नाहीये. So,
Exactly.
so if you don't have any baseline, whether these are the high, whether they are increasing or whether they are decreasing, you cannot come to know. So that is why it is recommended की patient आला आहे, you never know. असंही दिसलंय सेकंड फेज मध्ये की 30-40 वर्षाची मुलं आहेत आणि ती खाडकन जातात. सुरुवातीला असिम्प्टोमॅटिक आहे, दे हॅव नॉट टेकन प्रॉपर प्रिकॉशन आणि सडनली ते हे होत आहेत, कोलॅप्स व्हायला लागतात. And once the clot started forming, then it becomes difficult.
Ma'am, there is
treatment
one
असली
more.
बरी.
Yeah, ma'am, there is one more query. Like there are cases who are showing a response to lignocaine. You know, they had taken some lignocaine in for their treatment, and they developed a vasovagal phenomena with that lignocaine. So then they are asking whether they can go for vaccination or whether they'll get a similar attack. So how to go with this kind of patients?
See, first of all, such patients they need little counseling. Okay, and the lignocaine, or if they have allergy to any drug, they have to say that to the immunization center. Okay, so they will test them and then they will ask them to take the Covaxin.
Okay.
That's the thing they do.
Thank you so much.
Okay, ma'am, one more question. As per WHO India representative, India is an endemic stage. What are your views on this? And is the cases going down? Is silence before the storm?
It is very difficult to say, but it is expected that the case rate is coming down and down and down. So we, if it has been again the third wave or something like that, now we must have expected, we were expecting in the month of or end of October, we will see rise in cases because of the Ganpati and Dasra and Navratri. So we are just waiting for getting the Navratri over, and at the end of the October, if we start rising, seeing the rise in cases, then there might be a chance like little again high may come up, but the chances are very less so far whatever we have observed. Because initially what we have observed in the second wave in the March and April, the positivity rate was 28-30%. And yesterday when we had a video conference meeting with all the 90 labs in that state, the positivity rate of my own lab was 1.07%. So from 35%, it has now gone to 1.07. So it is expected like it will not come up again. And we always hope for the best.
Yes.
Yes.
Now, next question from Dr. Farasha. Many senior consultants, diagnostics were expecting third wave of COVID-19. Although recovery rate and incidence of COVID positive patient has reduced a lot, Ma'am, what are your views? I think we have answered already. Then next question, Dr. Prajot. As per who India represented? Okay, still repetitive. Now next question from Dr. Purti. Do our memory cells remember these values or virus when the patient has had previous COVID infection? Are reinfections always severe?
No. The memory cell are the one which keep the memory of that different antigens or the vaccine which we are given it. Okay. The same kind of memory cells are also formed when you have a natural infection. Okay. So you have the memory cells even after natural infection as well as after vaccination also. So that's the reason why if you have taken vaccine first and had infection also, so then you see later on the more number of antibody production or more number of memory cells like that. So when you have a memory cell, naturally the clinical manifestations will be less because they will immediately act on it and the antibodies or the immune response will be mounted and the clinical severity will be less and the duration of the illness will also be less.
One more question from Dr. Prajot. Is there any interaction reported of pediatric
Yes.
COVID vaccine versus the routine immunization vaccines? Is it safe?
See, pediatric vaccination, yesterday only they have declared like the same co-vaccine can be started and just last month only they have finished the trial with the pediatric cases. So we have no enough cases to see that whether they have any adverse event. But it is expected they will not have any adverse event with any vaccine because in the immunization is done maximally in the pediatric age group only. And it is just like any other vaccine. So there is, it is expected like there will not be any side effects as such. For the person who has asked a question regarding memory cell, has it convinced with the answer or has some different this thing?
Yes, ma'am.
Okay.
Okay. Anyone want to ask question? They can type their question. I think all questions have been answered and all of them are satisfied. So on behalf of Dr. G.D. Paul Foundation, YMT Homeopathic Medical College, I, Dr. Pallavi Chikle, Assistant Professor, Department of Pathology, take this opportunity to express my gratitude for all who have contributed directly and indirectly in this webinar. Today's day was really a day of learning. I would like to express my gratitude to today's guest speaker, Dr. Suvarna Joshi ma'am, for sparing her valuable time and enlightening us with knowledge. Ma'am, thank you for clearing our concepts and enhancing our understanding pertaining to the laboratory diagnosis of COVID-19 and myths of vaccination. I would like to thank Dr. G.D. Paul Sir, Chairman, Dr. G.D. Paul Foundation, YMT Homeopathic Medical College, for his valuable support. I would like to thank Dr. P.P. Page Ma'am, Principal, YMT Homeopathic Medical College, for her enthusiastic support. I would like to thank Dr. D.G. Bagal Sir, Vice Principal, YMT Homeopathic Medical College and Dean Faculty, MUHS Nashik, for his constant support. A big thanks to all participants for their active participation. I will thank teaching and non-teaching faculty for their unflinching support and contribution. Heartfelt thanks to the all organizing committee for the successful organization of this webinar. Thanks to our technical team, without which support this webinar wouldn't be possible. Then finally, with these warm words, we come to the conclusion of this webinar. Thank you so much.
Thank you so much, madam, for giving me opportunity to do the interaction with your people and the G.D. Paul Foundation. Thank you so much.